Institute for Advanced Studies, Jodhpur 32. Institute for Advanced Studies, B5. Peroxisomes. Peroxisomes are ubiquitous organelles in eukaryotes that participate in the metabolism of fatty acids and other metabolites. Peroxisomes have enzymes that rid the cell of toxic peroxides. They have a single lipid bilayer that separates their contents from the cytosol, the internal fluid of the cell, and contain membrane proteins critical for various functions, such as importing proteins into the organelles and aiding in proliferation. Lysosomes. Peroxisomes are part of the secretory pathway of a cell, but they are much more dynamic and can replicate by enlarging and then dividing. Peroxisomes were identified as cellular organelles by the Belgian cytologist Christian de Duve in 1967 after they had been first described in a Swedish PhD thesis a decade earlier. Occurrence and evolution. Peroxisomes are found in virtually all eukaryotic cells. Peroxisomes contain enzymes for certain oxidative reactions, like the beta oxidation of very long-chain fatty acids. Prokaryotes lack peroxisomes. The enzymatic content of peroxisomes varies across species, but the presence of certain proteins common to many species has been used to suggest an endosymbiotic origin. That is, peroxisomes evolved from bacteria that invaded larger cells as parasites, and very gradually evolve a symbiotic relationship. However, this view has been challenged by recent discoveries. For example, peroxisomellus mutans can restore peroxisomes. Upon introduction of the wild-type gene, and peroxisomes have been observed to be formed from the endoplasmic reticulum. An evolutionary analysis of the peroxisomal proteome found homologies between the peroxisomal import machinery and the ERAD pathway in the endoplasmic reticulum, along with a number of metabolic enzymes that were likely recruited from the mitochondria. 6. These results indicate that the peroxisome does not have an endosymbiotic origin. Instead, it likely originates from ER, and its proteins were recruited from pools existing within the primitive eukaryote, as quoted in the science textbook Biozone. Function. Peroxisomes contain oxidative enzymes, such as catalase, D-amino acid oxidase, and uric acid oxidase. Certain enzymes within the peroxisome, by using molecular oxygen, remove hydrogen atoms from specific organic substrates, labeled as R, in an oxidative reaction, producing hydrogen peroxide, H2O2, itself toxic. RH2 plus O2 R plus H2O2. Catalase, another enzyme in the peroxisome, in turn uses this H2O2 to oxidize other substrates, including phenols, formic acid, formaldehyde, and alcohol, by means of the peroxidation reaction, H2O2 plus RH2R plus 2H2O, thus eliminating the poisonous hydrogen peroxide in the process. This reaction is important in liver and kidney cells, where the peroxisomes detoxify various toxic substances that enter the blood. About 25% of the ethanol we drink is oxidized to acetaldehyde in this way. In addition, when excess H2O2 accumulates in the cell, catalase converts it to H2O through this reaction. 2H2O2 2H2O plus O2. A major function of the peroxisome is the breakdown of fatty acid molecules, in a process called beta oxidation. In this process, the fatty acids are broken down 2 carbons at a time, converted to acetyl-CoA, which is then transported back to the cytosol for further use. In animal cells, beta oxidation can also occur in the mitochondria. In yeast and plant cells, this process is exclusive for the peroxisome. The first reactions in the formation of plasmalogen in animal cells also occurs in peroxisomes. Plasmalogen is the most abundant phospholipid in myelin. Deficiency of plasmalogens causes profound abnormalities in the myelination of nerve cells, which is one of the reasons that many peroxisomal disorders lead to neurological disease. Peroxisomes also play a role in the production of bile acids and proteins. In higher plants, peroxisomes contain also a complex battery of antioxidative enzymes such as superoxide dismutase, the components of the ascorbate glutathione cycle, and the NADP dehydrogenases of the pentose phosphate pathway. It has been demonstrated the generation of superoxide O2 and nitric oxide NO radicals. They are also involved in photorespiration. In plants, special type of peroxisomes termed as glyoxysome carry out glyoxylate cycle in which lipids stored in seed are converted to carbohydrates and thus provide source of nutrition for developing embryo. Protein import. Proteins are selectively imported into peroxisomes. Since the organelles contain no DNA or ribosomes and thus have no means of producing proteins, all other proteins must be imported across the membrane. It is believed that necessary proteins enter through the endoplasmic reticulum during biogenesis as well as through membrane proteins. A specific protein signal, PTS or peroxisomal targeting signal, of 3 amino acids at the C-terminus of many peroxisomal proteins signals the membrane of the peroxisome to import them into the organelle. Other peroxisomal proteins contain a signal at the N-terminus. There are at least 32 known peroxisomal proteins, called peroxins, which participate in the process of importing proteins by means of ATP hydrolysis. Proteins do not have to unfold to be imported into the peroxisome. The protein receptors, the peroxins PEX5 and PEX7, accompany their cargoes containing a PTS1 or a PTS2, respectively, all the way into the peroxisome where they release the cargo and then return to the cytosol, a step named recycling. Overall, the import cycle is referred to as the extended shuttle mechanism. Evidence now indicates that ATP hydrolysis is required for the recycling of receptors to the cytosol. Also, ubiquitination appears to be crucial for the export of PEX5 from the peroxisome to the cytosol. Little is known about the import of PEX7, although it has helper proteins that have been shown to be ubiquitinated. Deficiencies. Peroxisomal disorders are a class of conditions that lead to disorders of lipid metabolism. One well-known example is Zellweger syndrome. Peroxisomes matrix proteins are synthesized on free ribosomes in the cytosol and that these proteins are imported post-translationally in pre-existing peroxisomes. Institute for Advanced Studies, Jodhpur 33. B6. Vacuoles. Vacuoles are found in the cytoplasm of most plant cells and some animal cells. Vacuoles are membrane-bound compartments within some eukaryotic cells that can serve a variety of secretory, excretory, and storage functions. Vacuoles and their contents are considered to be distinct from the cytoplasm, and are classified as ergastic according to some authors. Vacuoles are especially conspicuous in most plant cells. Functions. In general, vacuole functions include also Removing unwanted structural debris. Isolating materials that might be harmful or a threat to the cell. Containing waste products. Maintaining internal hydrostatic pressure or turgor within the cell. Maintaining an acidic internal pH. Containing small molecules. Exporting unwanted substances from the cell. Vacuoles also play a major role in autophagy, maintaining a balance between biogenesis, production, and degradation or turnover of many substances and cell structures. Vacuoles store food and other materials needed by a cell. They also aid in destruction of invading bacteria or of misfolded proteins that have begun to build up within the cell. The vacuole is a major part in the plant and animal cell. Protists. Some protists and macrophages use food vacuoles as a stage in phagocytosis, the intake of large molecules, particles, or even other cells, by the cell for digestion. They are also called storage sacs. A contractile vacuole is used to pump excess water out of the cell to reduce osmotic pressure and keep the cell from bursting, which is referred to as cytolysis or osmotic lysis. Budding yeast. In budding yeast cells, vacuoles act as storage compartments of amino acids and detoxification compartments. Under conditions of starvation, proteins are degraded in vacuoles. This is called autophagy. First, cytoplasms, mitochondrion, and small organelles are covered with multiplex plasma membranes called autophagosomes. Next, the autophagosomes fuse the vacuoles. Finally, the cytoplasms and the organelles are degraded. In a vacuole of budding yeast, black particles sometimes appear, called a dancing body. The dancing body moves actively in the vacuole and appears and disappears within 10 minutes to several hours. In previous research, it was suggested but not proven that the main component of the dancing body is polyphosphate acid. But the main component has been determined to be crystallized sodium polyphosphate and its function has been studied. It is thought that its function is to supply and store phosphates in budding yeast cells. Plants. Most mature plant cells have one or several vacuoles that typically occupy more than 30% of the cell's volume, and that can occupy as much as 90% of the volume for certain cell types and conditions. A vacuole is surrounded by a membrane called the tonoplast. This vacuole houses large amounts of a liquid called cell sap, composed of water, enzymes, inorganic ions like K+ and Cl, salts such as calcium, and other substances, including toxic byproducts removed from the cytosol to avoid interference with metabolism. Toxins present in the vacuole may also help to protect some plants from predators. Transport of protons from cytosol to vacuole aids in keeping cytoplasmic pH stable, while making the vacuolar interior more acidic, allowing degradative enzymes to act. Although having a large central vacuole is the most common case, the size and number of vacuoles may vary in different tissues and stages of development. Cells of the vascular cambium, for example, have many small vacuoles in winter, and one large one in summer. Aside from storage, the main role of the central vacuole is to maintain turgor pressure against the cell wall. Proteins found in the tonoplast control the flow of water into and out of the vacuole through active transport, pumping potassium K+ ions into and out of the vacuolar interior. Due to osmosis, water will diffuse into the vacuole, placing pressure on the cell wall. If water loss leads to a significant decline in turgor pressure, the cell will plasmolyze. Turgor pressure exerted by vacuoles is also helpful for cellular elongation, as the cell wall is partially degraded by the action of auxins. The less rigid wall is expanded by the pressure coming from within the vacuole. Vacuoles can help some plant cells to reach considerable size. Another function of a central vacuole is that it pushes all contents of the cell's cytoplasm against the cellular membrane, and thus keeps the chloroplasts closer to light. The vacuole also stores the pigments in flowers and fruits. Animals. Vacuoles in animals are a part of the processes of exocytosis and endocytosis. Exocytosis is the extrusion process of proteins from the Golgi apparatus initially enter secretory granules, where processing of prohormones to the mature hormones occurs before exocytosis, and also allows the animal cell to rid waste products. Endocytosis is the reverse of exocytosis. There are various types. Phagocytosis, cell eating, is the process by which bacteria, dead tissue, or other bits of material visible under the microscope are engulfed by cells. The material makes contact with the cell membrane, which then invaginates. The invagination is pinched off, leaving the engulfed material in the membrane-enclosed vacuole and the cell membrane intact. Pinocytosis, cell drinking, is essentially the same process, the difference being that the substances ingested are in solution and not visible under the microscope. Hydropic vacuolar changes are of importance of identifying various pathologies, such as the reversible cell swelling in renal tubules caused by hypoperfusion of the kidneys during open heart surgery. Institute for Advanced B7. Plastids. Plastids are major organelles found in plants and algae. Plastids often contain pigments used in photosynthesis, and the types of pigments present can change or determine the cell's color. Plastids are also the site of manufacture and storage of important chemical compounds used by the cell. Plastids in plants. Plastids are responsible for photosynthesis, storage of products like starch, and for the synthesis of many classes of molecules such as fatty acids and terpenes, which are needed as cellular building blocks and/or for the function of the plant. Depending on their morphology and function, plastids have the ability to differentiate, or redifferentiate, between these and other forms. All plastids are derived from proplastids, formerly eoplasts, don, early, which are present in the meristematic regions of the plant. Proplastids and young chloroplasts commonly divide, but more mature chloroplasts also have this capacity. In plants, plastids may differentiate into several forms, depending upon which function they need to play in the cell. Undifferentiated plastids, proplastids, may develop into any of the following plastids. Chloroplasts, for photosynthesis, see also etioplasts, the predecessors of chloroplasts. Chromoplasts, for pigment synthesis and storage. Leucoplasts, for monoterpene synthesis. Leucoplasts sometimes differentiate into more specialized plastids. Amyloplasts, for starch storage. Statoliths, for detecting gravity. Oleoplasts, for storing fat. Proteinoplasts, for storing and modifying protein. Each plastid creates multiple copies of the circular 75 to 250 kilobases plastid genome. The number of genome copies per plastid is flexible, ranging from more than 1,000 in rapidly dividing cells, which generally contain few plastids, to 100 or fewer in mature cells, where plastid divisions has given rise to a large number of plastids. The plastid genome contains about 100 genes encoding ribosomal and transfer ribonucleic acids, rRNAs and tRNAs, as well as proteins involved in photosynthesis and plastid gene transcription and translation. However, these proteins only represent a small fraction of the total protein setup necessary to build and maintain the structure and function of a particular type of plastid. Nuclear genes encode the vast majority of plastid proteins, and the expression of plastid genes in nuclear genes is tightly co-regulated to allow proper development of plastids in relation to cell differentiation. Plastid DNA exists as large protein DNA complexes associated with the Plastid DNA exists as large protein DNA complexes associated with the inner envelope membrane and called plastid nucleoids. Each nucleoid particle may contain more than 10 copies of the plastid DNA. The proplastid contains a single nucleoid located in the center of the plastid. The developing plastid has many nucleoids, localized at the periphery of the plastid, bound to the inner envelope membrane. During the development of proplastids to chloroplasts, and when plastids convert from one type to another, nucleoids change in morphology, size and location within the organelle. The remodeling of nucleoids is believed to occur by modifications to the composition and abundance of nucleoid proteins. Many plastids, particularly those responsible for photosynthesis, possess numerous internal membrane layers. In plant cells, long thin protuberances called stromoles sometimes form and extend from the main plastid body into the cytosol and interconnect several plastids. Proteins, and presumably smaller molecules, can move within stromoles. Most cultured cells that are relatively large compared to other plant cells have very long and abundant stromoles that extend to the cell periphery. Plastids in algae. In algae, the term leucoplast is used for all unpigmented plastids. Their function differ from the leucoplasts in plants. Etioplast, amyloplast and chromoplast are plant-specific and do not occur in algae. Algal plastids may also differ from plant plastids in that they contain pyrenoids. Inheritance of plastids. Most plants inherit the plastids from only one parent. Angiosperms generally inherit plastids from the mother, while many gymnosperms inherit plastids from the father. Algae also inherit plastids from only one parent. The plastid DNA of the other parent is thus completely lost. In normal interspecific crossings resulting in normal hybrids of one species, the inheritance of plastid DNA appears to be quite strictly 100% uniparental. In interspecific hybridizations, however, the inheritance of plastids appears to be more erratic. Although plastids inherit mainly maternally in interspecific hybridizations, there are many reports of hybrids of flowering plants that contain plastids of the father. Origin of plastids. Plastids are thought to have originated from endosymbiotic cyanobacteria. They developed around 1500 Maya and allowed eukaryotes to carry out oxygenic photosynthesis. Due to a split up into 3 evolutionary lineages, the plastids are named differently: chloroplasts in green algae and plants, rhodoplasts in red algae and cyanelles in the glaucophytes. The plastids differ by their pigmentation, but also in ultrastructure. The chloroplasts e.g. have lost all phycobilisms. The light harvesting complex is found in cyanobacteria, red algae and glaucophytes, but only in plants and in closely related green algae, contain stroma and granithylocoids. The Glaucocystophysin plastid, in contrast to the chloroplasts and the rhodoplasts, is still surrounded by remains of the Institute for Advanced Studies, Jodhpur 35. Cyanobacterial cell wall. All these primary plastids are surrounded by 2 membranes. Complex plastids start by secondary endosymbiosis, when a eukaryote engulfs a red or green alga and retains the algal plastid, which is typically surrounded by more than 2 membranes. In some cases these plastids may be reduced in their metabolic and/or photosynthetic capacity. Algae with complex plastids derived by secondary endosymbiosis of a red alga include the heteroconts, haptophytes, cryptomonids, and most dinoflagellates equals rhodoplasts. Those that endosymbiose a green alga include the euglenids and chlorarachniophytes equals chloroplasts. The Apicomplexa, a phylum of obligate parasitic protozoa including the causative agents of malaria, Plasmodium spp, Toxoplasmosis Toxoplasma gondii, and many other human or animal diseases also harbor a complex plastid, although this organelle has been lost in some apicomplexans, such as Cryptosporidium parvum, which causes cryptosporidiosis. The apicoplast is no longer capable of photosynthesis, but is an essential organelle, and a promising target for antiparasitic drug development. Some dinoflagellates take up algae as food and keep the plastid of the digested alga to profit from the photosynthesis. After a while the plastids are also digested. These captured plastids are known as kleptoplastids. B8. Chloroplast. Chloroplasts, the organelles responsible for photosynthesis, are in many respects similar to mitochondria. Both chloroplasts and mitochondria function to generate metabolic energy, evolved by endosymbiosis, contain their own genetic systems, and replicate by division. However, chloroplasts are larger and more complex than mitochondria, and they perform several critical tasks in addition to the generation of ATP. Most importantly, chloroplasts are responsible for the photosynthetic conversion of CO2 to carbohydrates. In addition, chloroplasts synthesize amino acids, fatty acids, and the lipid components of their own membranes. The reduction of nitrite NO2 to ammonia NH3, an essential step in the incorporation of nitrogen into organic compounds, also occurs in chloroplasts. Moreover, chloroplasts are only one of several types of related organelles, plastids, that play a variety of roles in plant cells. The structure and function of chloroplasts. Plant chloroplasts are large organelles 5 to 10 mm long that, like mitochondria, are bounded by a double membrane called the chloroplast envelope figure. In addition to the inner and outer membranes of the envelope, chloroplasts have a third internal membrane system, called the thylakoid membrane. The thylakoid membrane forms a network of flattened discs called thylakoids, which are frequently arranged in stacks called grana. Because of this three-membrane structure, the internal organization of chloroplasts is more complex than that of mitochondria. In particular, their three membranes divide chloroplasts into three distinct internal compartments. 1. The intermembrane space between the two membranes of the chloroplast envelope. 2. The stroma, which lies inside the envelope but outside the thylakoid membrane. And 3. The thylakoid lumen. Despite this greater complexity, the membranes of chloroplasts have clear functional similarities with those of mitochondria, as expected, given the role of both organelles in the chemiosmotic generation of ATP. The outer membrane of the chloroplast envelope, like that of mitochondria, contains porins and is therefore freely permeable to small molecules. In contrast, the inner membrane is impermeable to ions and metabolites, which are therefore able to enter chloroplasts only via specific membrane transporters. These properties of the inner and outer membranes of the chloroplast envelope are similar to the inner and outer membranes of mitochondria. In both cases, the inner membrane restricts the passage of molecules between the cytosol and the interior of the organelle. The chloroplast stroma is also equivalent in function to the mitochondrial matrix. It contains the chloroplast genetic system and a variety of metabolic enzymes, including those responsible for the critical conversion of CO2 to carbohydrates during photosynthesis. The major difference between chloroplasts and mitochondria, in terms of both structure and function, is the thylakoid membrane. This membrane is of central importance in chloroplasts, where it fills the role of the inner mitochondrial membrane in electron transport and the chemiosmotic generation of ATP Fig. The inner membrane of the chloroplast envelope, which is not folded into cristae, does not function in photosynthesis. Instead, the chloroplast electron transport system is located in the thylakoid membrane, and protons are pumped across this membrane from the stroma to the thylakoid lumen. The resulting electrochemical gradient then drives ATP synthesis as protons cross back into the stroma. In terms of its role in generation of metabolic energy, the thylakoid membrane of chloroplasts is thus equivalent to the inner membrane of mitochondria. The chloroplast genome. Like mitochondria, chloroplasts contain their own genetic system, reflecting their evolutionary origins from photosynthetic bacteria. The genomes of chloroplasts are similar to those of mitochondria in that they consist of circular DNA molecules present in multiple copies per organelle. However, chloroplast genomes are larger and Institute for Advanced Studies, Jodhpur 36. More complex than those of mitochondria, ranging from 120 to 160 kilobits and containing approximately 120 genes. Both the ribosomal and transfer RNAs used for translation of chloroplast mRNAs are encoded by the organelle genome. These include 4 rRNAs, 23S, 16S, 5S, and 4.5S and 30 tRNA species. In contrast to the smaller number of tRNAs encoded by the mitochondrial genome, the chloroplast tRNAs are sufficient to translate all the mRNA codons according to the universal genetic code. In addition to these RNA components of the translation system, the chloroplast genome encodes about 20 ribosomal proteins, which represent approximately a third of the proteins of chloroplast ribosomes. Some subunits of RNA polymerase are also encoded by chloroplasts, although additional RNA polymerase subunits and other factors needed for chloroplast gene expression are encoded in the nucleus. The chloroplast genome also encodes approximately 30 proteins that are involved in photosynthesis, including components of photosystems I and II, of the cytochrome Bf complex, and of ATP synthase. In addition, one of the subunits of ribulose bisphosphate carboxylase, Rubisco, is encoded by chloroplast DNA. Rubisco is the critical enzyme that catalyzes the addition of CO2 to ribulose 1,5-bisphosphate during the Calvin cycle. Not only is it the major protein component of the chloroplast stroma, but it is also thought to be the single most abundant protein on Earth, so it is noteworthy that one of its subunits is encoded by the chloroplast genome. B9. Structure and function of cytoskeleton and its role in motility. The membrane-enclosed organelles discussed in the preceding section constitute one level of the organizational substructure of eukaryotic cells. A further level of organization is provided by the cytoskeleton, which consists of a network of protein filaments extending throughout the cytoplasm of all eukaryotic cells. The cytoskeleton provides a structural framework for the cell, serving as a scaffold that determines cell shape and the general organization of the cytoplasm. In addition to playing this structural role, the cytoskeleton is responsible for cell movements. These include not only the movements of entire cells, but also the internal transport of organelles and other structures, such as meiotic chromosomes, through the cytoplasm. Importantly, the cytoskeleton is much less rigid and permanent than its name implies. Rather, it is a dynamic structure that is continually reorganized as cells move and change shape, for example, during cell division. The cytoskeleton is composed of three principal types of protein filaments: actin filaments, intermediate filaments, and microtubules, which are held together and linked to subcellular organelles in the plasma membrane by a variety of accessory proteins. This chapter discusses the structure and organization of each of these three major components of the cytoskeleton, as well as their roles in cell motility, organelle transport, cell division, and other types of cell movements. Structure and organization of actin filaments. The major cytoskeletal protein of most cells is actin, which polymerizes to form actin filaments thin, flexible fibers approximately 7 nanometers in diameter and up to several micrometers in length. Within the cell, actin filaments, also called microfilaments, are organized into higher order structures, forming bundles or three-dimensional networks with the properties of semisolid gels. The assembly and disassembly of actin filaments, their cross-linking into bundles and networks, and their association with other cell structures, such as the plasma membrane, are regulated by a variety of actin-binding proteins, which are critical components of the actin cytoskeleton. Actin filaments are particularly abundant beneath the plasma membrane, where they form a network that provides mechanical support, determines cell shape, and allows movement of the cell surface, thereby enabling cells to migrate, engulf particles, and divide. Individual actin molecules are globular proteins of 375 amino acids, 43 kb. Each actin monomer, globular G actin, has tight binding sites that mediate head-to-tail interactions with two other actin monomers, so actin monomers polymerize to form filaments filamentous F actin. Each monomer is rotated by 166 O in the filaments, which therefore have the appearance of a double-stranded helix. Because all the actin monomers are oriented in the same direction, actin filaments have a distinct polarity and their ends, called the plus and minus ends, are distinguishable from one another. It is noteworthy that several drugs useful in cell biology act by binding to actin and affecting its polymerization. For example, the cytochastolins bind to the plus ends of actin filaments and block their elongation. This results in changes in cell shape as well as inhibition of some types of cell movements, e.g., cell division following mitosis, indicating that actin polymerization is required for these processes. Another drug, phalloidin, binds tightly to actin filaments and prevents their dissociation into individual actin molecules. Phalloidin labeled with a fluorescent dye is frequently used to visualize actin filaments by fluorescence microscopy. Intermediate filaments. Intermediate filaments have a diameter of about 10 nanometers, which is intermediate between the diameters of the two other principal elements of the cytoskeleton, actin filaments about 7 nanometers and microtubules about 25 nanometers. In contrast to actin filaments and microtubules, the intermediate filaments are not directly involved in cell movements. Instead, they appear to play basically a structural role by providing mechanical strength to cells and tissues. Intermediate filament proteins. Whereas actin filaments and microtubules are polymers of single types of proteins, actin and tubulin, respectively, intermediate filaments are composed of a variety of proteins that are expressed in different types of cells. More than 50 different intermediate filament proteins have been identified and classified into 6 groups based on similarities between their amino acid sequences. Institute for Advanced Studies, Jodhpur 37. Types I and II consist of 2 groups of keratins, each consisting of about 15 different proteins, which are expressed in epithelial cells. Some type 1 and 2 keratins, called hard keratins, are used for production of structures such as hair, nails, and horns. The type 3 intermediate filament proteins include vimentin, which is found in a variety of different kinds of cells, including fibroblasts, smooth muscle cells, and white blood cells. Another type 3 protein, desmin, is specifically expressed in muscle cells, where it connects the Z discs of individual contractile elements. The type 4 intermediate filament proteins include the 3 neurofilament NF proteins designated NFL, NFM, and NFH for light, medium, and heavy, respectively. These proteins form the major intermediate filaments of many types of mature neurons. The type 5 intermediate filament proteins are the nuclear lamins, which are found in most eukaryotic cells. Rather than being part of the cytoskeleton, the nuclear lamins are components of the nuclear envelope. They also differ from the other intermediate filament proteins in that they assemble to form an orthogonal meshwork underlying the nuclear membrane. Despite considerable diversity in size and amino acid sequence, the various intermediate filament proteins share a common structural organization. Microtubules. Microtubules, the third principal component of the cytoskeleton, are rigid hollow rods approximately 25 nanometers in diameter. Like actin filaments, microtubules are dynamic structures that undergo continual assembly and disassembly within the cell. They function both to determine cell shape and in a variety of cell movements, including some forms of cell locomotion, the intracellular transport of organelles, and the separation of chromosomes during mitosis. Structure, assembly, and dynamic instability of microtubules. In contrast to intermediate filaments, which are composed of a variety of different fibrous proteins, microtubules are composed of a single type of globular protein, called tubulin. Tubulin is a dimer consisting of 2 closely related 55 kD polypeptides, alpha tubulin and beta tubulin. Like actin, both alpha and beta tubulin are encoded by small families of related genes. In addition, a third type of tubulin, gamma tubulin, is specifically localized to the centrosome, where it plays a critical role in initiating microtubule assembly. Tubulin dimers polymerize to form microtubules, which generally consist of 13 linear protofilaments assembled around a hollow core. The protofilaments, which are composed of head-to-tail arrays of tubulin dimers, are arranged in parallel. Consequently, microtubules, like actin filaments, are polar structures with 2 distinct ends, a fast-growing plus end and a slow-growing minus end. This polarity is an important consideration in determining the direction of movement along microtubules, just as the polarity of actin filaments defines the direction of myosin movement. Tubulin dimers can depolymerize as well as polymerize, and microtubules can undergo rapid cycles of assembly and disassembly. Both alpha and beta tubulin bind GTP, which functions analogously to the ATP bound to actin to regulate polymerization. In particular, the GTP bound to B tubulin, though not that bound to A tubulin, is hydrolyzed to GDP during or shortly after polymerization. Whether a microtubule grows or shrinks is determined by the rate of tubulin addition relative to the rate of GTP hydrolysis. C1. Organization of genes. In molecular terms, a gene commonly is defined as the entire nucleic acid sequence that is necessary for the synthesis of a functional polypeptide. According to this definition, a gene includes more than the nucleotides encoding the amino acid sequence of a protein, referred to as the coding region. A gene also includes all the DNA sequences required for synthesis of a particular RNA transcript. Thus, a gene is the entire DNA sequence required for synthesis of a functional protein or RNA molecule. In addition to the coding regions, exons, a gene includes transcription control regions and sometimes introns. Although the majority of genes encode proteins, some encode tRNAs, rRNAs, and other types of RNA. Most bacterial genes have no introns, whereas most genes of multicellular organisms do. The introns in human genes encoding average-size proteins are often much longer than the exons. Many bacterial proteins with related functions are encoded by contiguous genes regulated by a single transcription control region. This type of gene cluster, called an operon, is transcribed into a single, polycistronic mRNA, which is translated to yield several different proteins. Most eukaryotic transcription units are transcribed into monocistronic mRNAs, each of which is translated into a single protein. The primary transcript produced from a simple eukaryotic transcription unit is processed into a single type of mRNA. The primary transcript produced from a complex eukaryotic transcription unit can be processed into 2 or more different mRNAs depending on the choice of splice sites and/or polyadenylation site. In the case of many complex units, one mRNA is produced in one cell type, while an alternative mRNA is produced in a different cell type. Organization of genes and non-coding DNA. The genomes of prokaryotes and lower eukaryotes contain few non-functional sequences, whereas vertebrate genomes contain many sequences that do not code for RNAs or have any structural or regulatory function. Only about 5% of the genomic DNA in humans encodes proteins or functional RNAs. The lack of a consistent relationship between the amount of DNA in the haploid chromosomes of an animal or plant and its phylogenetic complexity is called the C-value paradox. The human genome. This term is used to describe the different types of sequence that together make up the DNA in a human cell. The DNA in the human genome is about 3 billion base pairs (BP) long and is estimated to contain 30,000 genes. The DNA is arranged as a set of 23 chromosomes, each of which is a single, double-stranded DNA molecule 55 to 250 million BP long. The genes and gene-related sequences account for about 25% of the DNA Fig. The remainder is called extragenic DNA and has no known function. Genes and gene-related sequences, 20-30%. Genes. The coding information in a gene is present as a series of segments of DNA sequence called exons separated from each other by intervening non-coding sequences called introns. Genes vary greatly in size and also with respect to the number and sizes of the introns. Some genes such as the histone H4 gene are just a few hundred base pairs long. Others, such as the factor VIII gene, are several hundred kilobase pairs (kbp) in length and contain many large introns such that the actual coding sequence accounts for just a few % of the total gene sequence. Additional sequences are present which are associated with genes. Leader and trailer sequences occur at the 5' and 3' ends of the gene which are transcribed but not translated. Promoter sequences occur upstream of the point where transcription begins and regulates synthesis of mRNA from the gene. The promoter may extend up to about 1 kbp upstream, but other regulatory sequences that influence transcription may occur at sites much further away. Gene families. Some genes exist as a number of copies with identical or related sequences that can be grouped into families. Gene families may be organized in a number of ways. All of the genes in the family occur at the same chromosomal locus. An example of this is the growth hormone gene family, whose 5 members are clustered on chromosome 17. The genes belonging to the family may occur at different loci. For example, the 5 members of the aldolase gene family are on different chromosomes. The genes of a family may exist as a series of clusters on different chromosomes. An example is the homeobox genes, which occur as 4 clusters on separate chromosomes, each containing about 10 individual genes. In some multigene families, all the genes are identical and may encode a protein required in large amounts by the cell, such as histones. In other families, the genes are not identical but show some sequence divergence. In some cases, the divergence is so great that the genes encode proteins that are related but have distinctive properties. An example of this is the alpha and beta globin gene families, whose members are expressed at different stages of embryonic development and in adults. Pseudogenes. These are diverged members of gene families that have acquired one or more inactivating mutations so that they are no longer able to function and do not produce biologically active protein. A pseudogene is simply a mutated version of a parent gene. Often the mutations present are nonsense mutations which generate stop codons and result in premature termination of translation. Related to pseudogenes are processed pseudogenes, which are DNA copies of mRNA. Processed pseudogenes lack introns, and because they do not have a promoter, they are not transcribed and do not result in protein production. It is not clear how processed pseudogenes arose in the human genome, but they may have been derived by reverse transcription of mRNA into double-stranded DNA, which was then inserted into the genome. Another group of inactive genes are gene fragments which lack the 5' or 3' region of the parent gene. These are thought to have arisen by a deletion event or by recombination that split the parent gene. Extragenic DNA, 70-80%. This part of the human genome is composed of sequences that exist in addition to the genes and gene-related sequences described above. Fig. Extragenic DNA is composed of sequences that are not part of a gene, exons and introns, not associated with a gene, leader and trailer sequences, promoters and distant regulatory elements, and not a pseudogene or a gene fragment. Although extragenic sequences account for most of the DNA in the human genome, 70-80%, they have no known function. Most of the extragenic DNA sequences, 70-80%, are unique or exist as a small number of copies. The remainder, 20-30%, are moderately or highly repeated sequences that may be dispersed throughout the genome or lined up end on end as long tandem arrays. Dispersed repetitive sequences. 2 types of dispersed repetitive sequence exist, known as short and long interspersed nuclear elements, abbreviated to SINEs and lines. SINEs. The best known example of SINEs is the Lu elements. These sequences are not identical, but they are similar enough to be classed as a family. They have an average length of 250 BP and almost 1 million copies exist, which are very widely dispersed throughout the genome, occurring in most places including the introns of some genes. The origin of Lu elements is uncertain, but they are believed to have arisen as one or more processed pseudogenes. It is suggested that at one stage these sequences acquired transposable activity which allowed them to replicate and move to different sites in the genome. Lines. They are similar to SINEs but have longer sequences. A well-known example is the L1 line, which is 6,500 BP long and is present as 60,000 copies. Lines are a type of retroelement. These are sequences that are capable of moving through the genome by a process called transposition, which allows them to copy themselves by reverse transcription and to insert the copy into the genome at a distant site. Fig. Most L1 elements are truncated, but the full-length version has all the components needed for transposition, including a gene that could code for reverse. Clustered repetitive sequences. The human genome contains extensive regions in which repetitive sequences are arranged end on end as long tandem arrays. This is called satellite DNA and occurs in various forms, each with a different repeat unit. Because they are separated from the bulk of cellular DNA by equilibrium density gradient centrifugation. However, not all simple sequence DNA is separate from the bulk of cellular DNA during centrifugation. Simple sequence DNA, which consists largely of very short sequences repeated in long tandem arrays, is preferentially located in centromeres, telomeres, and specific locations within the arms of particular chromosomes where it may have a structural role. Three classes of satellite DNA have been defined based on size of the cluster and the length of the repeat unit. Satellite DNA. Satellite DNA was the first type to be identified in the human genome and contains repeat units of up to 200 BP arranged in clusters between 100 and 5000 kbp. Minisatellite DNA. Minisatellite DNA occurs in smaller clusters up to 20 kbp in length and has repeat units up to 25 BP. The length of a particular simple sequence tandem array is quite variable among individuals in a species, probably because of unequal crossing over during meiosis. Differences in the lengths of minisatellite DNA forms the basis for DNA fingerprinting. Microsatellite DNA. Microsatellite DNA occurs in short clusters, usually less than 150 BP, and has a repeat unit which is usually 4 BP or less. Microsatellite DNA is very common. One type, which contains the dinucleotide CA as the repeat sequence, accounts for 0.5% of the entire genome. Mononucleotide repeats, consisting of a single repeated base, account for a further 0.3%. Variable number of tandem repeats. The length of microsatellite DNA sequences such as the CA repeats and minisatellite DNA varies from person to person. At any given location in the genome, the number of repeat units present in an individual microsatellite DNA sequence may vary by as many as 10 or more. As such, these sequences are known as variable number tandem repeats or VNTRs. The polymerase chain reaction (PCR) can be used to analyze VNTRs Fig. Using primers specific for unique sequences on either side of the repeat sequence, DNA molecules of varying lengths are amplified depending on how many repeats are present. By examining several VNTRs, it is possible to build up a genetic profile that is unique to the individual being. VNTRs have a number of very useful applications based on establishing the identity of an individual. They can be used in forensic science to link suspects to a crime using biological material recovered from the scene. They also have a number of uses in medicine, including matching patients undergoing transplants with donor organs, identifying carriers of genetic diseases, and establishing paternity. VNTRs have also been widely used in the Human Genome Mapping Project as gene markers to identify the location and order of genes on chromosomes. C2. Genome organization. Bacterial chromosomes. In most bacterial cells, genes are encoded on large circular chromosomes. The circular nature of bacterial chromosomes was first discovered by analyzing the frequency of genetic recombination between mutant genes that produced easily assayed phenotypes, such as the inability to grow in the absence of a specific amino acid or the inability to grow on a particular sugar. Eventually, the circular structure of the E. coli chromosome was observed directly in autoradiographs of DNA molecules from cells grown in the presence of 3-labelled thymine, which is incorporated only into DNA. Such studies showed that the E. coli chromosome has a total length of 1 mm. The E. coli chromosomal DNA molecule replicates from a single replication origin, similar to the small circular plasmid DNA molecule. The 1 mm long DNA molecule of the E. coli chromosome is contained within cells that are only about 2 micrometers long and about 0.51 micrometer wide. A free DNA molecule of this size would form a random coil about 1,000 times the volume of an E. coli cell. However, several mechanisms operate to compact E. coli chromosomal DNA sufficiently to fit inside the bacterial cell. For example, the large volume filled by free DNA is due largely to charge repulsion between the negatively charged phosphate groups. In the cell, this effect is counteracted by association of the DNA with positively charged polyamines, such as spermine and spermidine, which shield the negative charges of the DNA phosphate groups. In addition, numerous small protein molecules associate with the chromosomal DNA, causing it to fold into a more compact structure. The most abundant of these proteins, HNS, is a dimer of a 15.6-kDa polypeptide. HNS binds DNA tightly and compacts it considerably, as indicated by the increased sedimentation rate and decreased viscosity of DNA associated with HNS compared with free DNA. There are about 20,000 HNS molecules per E. coli cell, enough for one HNS dimer per 400 base pairs of DNA. Finally, E. coli chromosomal DNA is tightly supercoiled, that is, twisted upon itself like the circular SV40 DNA. An E. coli enzyme called DNA gyrase uses energy from ATP hydrolysis to wind supercoils into DNA. Supercoiling contributes to the compaction necessary to fit chromosomal DNA into the bacterial cell. If all the supercoils were relaxed and the DNA spread out, it would appear as a single, replicating, circular DNA molecule. Eukaryotic nuclear DNA associates with histone proteins to form chromatin. The most abundant proteins associated with eukaryotic DNA are histones, a family of basic proteins present in all eukaryotic nuclei. The 5 major types of histone proteins, termed H1, H2A, H2B, H3, and H4, are rich in positively charged basic amino acids, which interact with the negatively charged phosphate groups in DNA. In a fraction of the histone proteins of most cells, some of the basic amino acid side chains are modified by post-translational addition of acetyl CH3COO phosphate, or methyl groups, neutralizing the positive charge of the side chain or converting it to a negative charge. The amino acid sequences of 4 histones H2A, H2B, H3, and H4 are remarkably similar among distantly related species. For example, the sequences of histone H3 from sea urchin tissue and of H3 from calf thymus are identical except for a single amino acid, and only 4 amino acids are different in H3 from the garden pea and that from calf thymus. Minor histone variants encoded by genes that differ from the highly conserved major types also exist, particularly in vertebrates. The amino acid sequence of H1 varies more from organism to organism than do the sequences of the other major histones. In certain tissues, H1 is replaced by special histones. For example, in the nucleated red blood cells of birds, a histone termed H5 is present in place of H1. The similarity in sequence among histones from all eukaryotes indicates that they fold into very similar three-dimensional conformations, which were optimized for histone function early in evolution in a common ancestor of all modern eukaryotes. Chromatin. The total length of DNA in eukaryotic cell depends on the species, but it can be thousands of times as much as in a prokaryotic genome, and is made up of a number of discrete bodies called chromosomes, 46 in humans. The DNA in each chromosome is believed to be a single linear molecule, which can be up to several centimeters long. This entire DNA must be packaged into the nucleus, a space of approximately the same volume as a bacterial cell. In fact, in their most highly condensed forms, the chromosomes have an enormously high DNA concentration of perhaps 200 mg/ml1. This feat of packing is accomplished by the formation of a highly organized complex of DNA and protein, known as chromatin, a nucleoprotein complex. More than 50% of the mass of chromatin is protein. Chromosomes greatly alter their level of compactness as cells progress through the cell cycle, varying between highly condensed chromosomes at metaphase just before cell division and very much more diffuse structures in interphase. This implies the existence of different levels of organization of chromatin. Histones. Most of the protein in eukaryotic chromatin consists of histones, of which there are 5 families, or classes, H2A, H2B, H3 and H4, known as the core histones, and H1. The core histones are small proteins, with masses between 10 and 20 kDa, and H1 histones are a little larger at around 23 kDa. All histone proteins have a large positive charge. Between 20 and 30% of their sequences consist of the basic amino acids, lysine and arginine. This means that histones will bind very strongly to the negatively charged DNA in forming chromatin. Members of the same histone class are very highly conserved between relatively unrelated species, for example between plants and animals, which testifies to their crucial role in chromatin. Within a given species, there are normally a number of closely similar variants of a particular class, which may be expressed in different tissues and at different stages in development. There is not much similarity in sequence between the different histone classes, but structural studies have shown that the classes do share a similar tertiary structure, suggesting that all histones are ultimately evolutionarily related. H1 histones are somewhat distinct from the other histone classes in a number of ways. In addition to their larger size, there is more variation in H1 sequences both between and within species than in the other classes. Histone H1 is more easily extracted from bulk chromatin, and seems to be present in roughly half the quantity of the other classes, of which there are very similar amounts. These facts suggest a specific and distinct role for histone H1 in chromatin structure. Nucleosomes. A number of studies in the I-970s pointed to the existence of a basic unit of chromatin structure. Nucleases are enzymes which hydrolyze the phosphodiester bonds of nucleic acids. Exonucleases release single nucleotides from the ends of nucleic acid strands, whereas endonucleases cleave internal phosphodiester bonds. Treatment of chromatin with micrococcal nuclease, an endonuclease which cleaves double-stranded DNA, led to the isolation of DNA fragments with discrete sizes in multiples of approximately 200 bp. It was discovered that each 200 bp fragment is associated with an octamer core of histone proteins, H2A2, H2B2, H3, H4, which is why these are designated the core histones, and more loosely with one molecule of H1. The proteins protect the DNA from the action of micrococcal nuclease. More prolonged digestion with nuclease leads to the loss of HL and yields a very resistant structure consisting of 146 bp of DNA associated very tightly with the histone octamer. This structure is known as the nucleosome core, and is structurally very similar whatever the source of the chromatin. The structure of the nucleosome core particle is now known in considerable detail, from structural studies culminating in X-ray crystallography. The histone octamer forms a wedge-shaped disc, around which the 146 bp of DNA is wrapped in 1.8 turns in a left-handed direction. Figure 1 shows the basic features and the dimensions of the structure. The left-handed wrapping of the DNA around the nucleosome corresponds to negative supercoiling, that is, the turns are superhelical turns. Although eukaryotic DNA is negatively supercoiled to a similar level as that of prokaryotes, on average, virtually all the supercoiling is accounted for by wrapping in nucleosomes and there is no unconstrained supercoiling. The role of H1, one molecule of histone Hi binds to the nucleosome, and acts to stabilize the point at which the DNA enters and leaves the nucleosome core. In the presence of HI, a further 20 bp of DNA is protected from nuclease digestion, making 166 bp in all, corresponding to 2 full turns around the histone octamer. A nucleosome core plus Hi is known as a chromatosome. The larger size of HI compared with the core histones is due to the presence of an additional C-terminal tail, which serves to stabilize the DNA between the nucleosome cores. As stated above, H1 is more variable in sequence than the other histones and, in some cell types, it may be replaced by an extreme variant called histone H5, which binds chromatin particularly tightly, and is associated with DNA which is not undergoing transcription. Linker DNA, in electron micrographs of nucleosome core particles on DNA under certain conditions, an array, sometimes called the beads on a string, structure is visible. This comprises globular particles, nucleosomes connected by thin strands of DNA. This linker DNA is the additional DNA required to make up the 200 bp nucleosomal repeat apparent in the micrococcal nuclease experiments, see above. The average length of linker DNA between core particles is 55 bp, but the length varies between species and tissues from almost nothing to more than 100 bp. The solenoid 30 nanometers fiber. The presence of histone Hi increases the organization of the beads on a string, to show a zig-zag structure in electron micrographs. With a change in the salt concentration, further organization of the nucleosomes into a fiber of 30 nanometers diameter takes place. Detailed studies of this process have suggested that the nucleosomes are wound into a higher-order left-handed helix, dubbed a solenoid, with around 6 nucleosomes per turn. However, there is still some conjecture about the precise organization of the fiber structure, including the path of the linker DNA and the way in which different linker lengths might be incorporated into what seems to be a very uniform structure. Most chromosomal DNA in vivo is packaged into the 30 nanometers fiber. Higher-order structure. The organization of chromatin at the highest level seems rather similar to that of prokaryotic DNA. Electron micrographs of chromosomes which have been stripped of their histone proteins show a looped domain structure. Even the size of the loops is approximately the same, up to around 100 kilobits of DNA, although there are many more loops in a eukaryotic chromosome. The loops are constrained by interaction with a protein complex known as the nuclear matrix. The DNA in the loops is in the form of 30 nanometers fiber, and the loops form an array about 300 nanometers across. C3. Transposons. Mobile DNA. The second type of repetitious DNA in eukaryotic genomes, termed interspersed repeats, also known as moderately repeated DNA, or intermediate repeat DNA, is composed of a very large number of copies of relatively few sequence families. These sequences, which are interspersed throughout mammalian genomes, make up approximately equals 25 to 50% of mammalian DNA, approximately equals 45% of human DNA. Because moderately repeated DNA sequences have a unique ability to move in the genome, they are called mobile DNA elements or transposable elements. Although mobile DNA elements, ranging from hundreds to a few thousand base pairs in length, originally were discovered in eukaryotes, they also are found in prokaryotes. The process by which these sequences are copied and inserted into a new site in the genome is called transposition. Mobile DNA elements, or simply mobile elements, are essentially molecular symbiotes that in most cases appear to have no specific function in the biology of their host organisms, but exist only to maintain themselves. For this reason, Francis Crick referred to them as selfish DNA. When transposition of eukaryotic mobile elements occurs in germ cells, the transposed sequences of their new sites can be passed on to succeeding generations. In this way, mobile elements have multiplied and slowly accumulated in eukaryotic genomes over evolutionary time. Since mobile elements are eliminated from eukaryotic genomes very slowly, they now constitute a significant portion of the genomes of many eukaryotes. Transposition also may occur within a somatic cell. In this case, the transposed sequence is transmitted only to the daughter cells derived from that cell. In rare cases, this may lead to a somatic cell mutation with detrimental phenotypic effects, for example, the inactivation of a tumor suppressor gene. Movement of mobile elements involves a DNA or an RNA intermediate. Barbara McClintock discovered the first mobile elements while doing classical genetic experiments in maize corn during the 1940s. She characterized genetic entities that could move into and back out of genes, changing the phenotype of corn kernels. Her theories were very controversial until similar mobile elements were discovered in bacteria, where they were characterized as specific DNA sequences, and the molecular basis of their transposition was deciphered. As research on mobile elements progressed, they were found to fall into two categories: 1, those that transpose directly as DNA, and 2, those that transpose via an RNA intermediate transcribed from the mobile element by an RNA polymerase and then converted back into double-stranded DNA by a reverse transcriptase, figure 1. Mobile elements that transpose through a DNA intermediate are generally referred to as DNA transposons. Mobile elements that transpose to new sites in the genome via an RNA intermediate are called retrotransposons because their movement is analogous to the infectious process of retroviruses. Indeed, retroviruses can be thought of as retrotransposons that evolve genes encoding viral coats, thus allowing them to transpose between cells. Retrotransposons can be further classified on the basis of their specific mechanism of transposition. Mobile elements that move as DNA are present in prokaryotes and eukaryotes. Most mobile elements in bacteria transpose directly as DNA. In contrast, most mobile elements in eukaryotes are retrotransposons, but eukaryotic DNA transposons also occur. Indeed, the original mobile elements discovered by Barbara McClintock are DNA transposons. Bacterial insertion sequences. The first molecular understanding of mobile elements came from the study of certain E. coli mutations caused by the spontaneous insertion of a DNA sequence, approximately equals 1 to 2 kb long, into the middle of a gene. These inserted stretches of DNA are called insertion sequences, or as elements. So far, more than 20 different as elements have been found in E. coli and other bacteria. Transposition of an as element is a very rare event, occurring in only 1 in 1,0,5,1,0,7 cells per generation, depending on the as element. Many transpositions inactivate essential genes, killing the host cell and the as elements it carries. Therefore, higher rates of transposition would probably result in too great a mutation rate for the host cell to survive. However, since as elements transpose more or less randomly, some transposed sequences enter non-essential regions of the genome, e.g., regions between genes, allowing the cell to survive. At a very low rate of transposition, most host cells survive and therefore propagate the symbiotic as element. As elements also can insert into plasmids or lysogenic viruses, and thus be transferred to other cells. When this happens, as elements can transpose into the chromosomes of virgin cells. The general structure of as elements is diagrammed in figure 2. An inverted repeat, usually containing approximately equals 50 base pairs, invariably is present at each end of an insertion sequence. In an inverted repeat, the 5'3" sequence on one strand is repeated on the other strand, as. Between the inverted repeats is a region that encodes transposase, an enzyme required for transposition of the as element to a new site. The transposase is expressed at a very low rate, accounting for the very low frequency of transposition. An important hallmark of as elements is the presence of a short direct repeat sequence, containing 5 to 11 base pairs, immediately adjacent to both ends of the inserted element. The length of the direct repeat is characteristic of each type of as element, but its sequence depends on the target site where a particular copy of the as element is inserted. When the sequence of a mutated gene containing an as element is compared with the sequence of the wild-type gene before insertion, only one copy of the short direct repeat sequence is found in the wild-type gene. Duplication of this target site sequence to create the second direct repeat adjacent to an as element occurs during the insertion process. As depicted in figure 3, transposition of an as element is similar to a cut-and-paste operation in a word processing program. Transposase performs 3 functions in this process. It 1. Precisely excises the as element in the donor DNA, 2. Makes staggered cuts in a short sequence in the target DNA, and 3. Ligates the 3' termini of the as element to the 5' ends of the cut donor DNA. Finally, a host cell DNA polymerase fills in the single-stranded gaps, generating the short direct repeats that flank as elements, and DNA ligase joins the free ends. Eukaryotic DNA transposons. McClintock's original discovery of mobile elements came from observation of certain spontaneous mutations in maize that affect production of any of the several enzymes required to make anthocyanin, a purple pigment in maize kernels. Mutant kernels are white, and wild-type kernels are purple. One class of these mutations is revertible at high frequency, whereas a second class of mutations does not revert unless they occur in the presence of the first class of mutations. McClintock called the agent responsible for the first class of mutations the activator AC element and those responsible for the second class dissociation Ds elements because they also tended to be associated with chromosome breaks. Many years after McClintock's pioneering discoveries, cloning and sequencing revealed that AC elements are equivalent to bacterial as elements. Like as elements, they contain inverted terminal repeat sequences that flank the coding region for a transposase, which recognizes the terminal repeats and catalyzes transposition to a new site in DNA. These elements are deleted forms of the AC element in which a portion of the sequence encoding transposase is missing. Because it does not encode a functional transposase, a Ds element cannot move by itself. However, in plants that carry the AC element, and thus express a functional transposase, Ds elements can move. Since McClintock's early work on mobile elements in corn, transposons have been identified in other eukaryotes. For instance, approximately half of all the spontaneous mutations observed in Drosophila are due to the insertion of mobile elements. Although most of the mobile elements in Drosophila function as retrotransposons, at least one, the P element, functions as a DNA transposon, moving by a cut and paste mechanism similar to that used by bacterial insertion sequences. Current methods for constructing transgenic Drosophila depend on engineered, high-level expression of the P element transposase and use of the Pellement inverted terminal repeats as targets for transposition. DNA transposition by the cut and paste mechanism can result in an increase in the copy number of a transposon when it occurs during S phase, the period of the cell cycle when DNA synthesis occurs. This happens when the donor DNA is from one of the two daughter DNA molecules in a region of a chromosome that has replicated and the target DNA is in the region that has not yet replicated. When DNA replication is complete at the end of the S phase, the target DNA in its new location is also replicated. This results in a net increase by 1 in the total number of these transposons in the cell. When this occurs during the S phase preceding meiosis, 2 of the 4 germ cells produced have the extra copy. Repetition of this process over evolutionary time has resulted in the accumulation of large numbers of DNA transposons in the genomes of some organisms. Human DNA contains about 300,000 copies of full-length and deleted DNA transposons, amounting to approximately equals 3% of human DNA. Some retrotransposons contain LTRs and behave like intracellular retroviruses. The genomes of all eukaryotes studied from yeast to humans contain retrotransposons, mobile DNA elements that transpose through an RNA intermediate utilizing a reverse transcriptase, see Figure 1b. These mobile elements are divided into 2 major categories, those containing and those lacking long terminal repeats, LTRs. LTR retrotransposons, which we discuss in this section, are common in yeast, e.g., Ti elements, and in Drosophila, e.g., Copia elements. Although less abundant in mammals than non-LTR retrotransposons, LTR retrotransposons nonetheless constitute approximately equals 8% of human genomic DNA. Because they exhibit some similarities with retroviruses, these mobile elements sometimes are called viral retrotransposons. In mammals, retrotransposons lacking LTRs are the most common type of mobile element. The general structure of LTR retrotransposons found in eukaryotes is depicted in Figure 4. In addition to short 5' and 3' direct repeats typical of all mobile elements, these retrotransposons are marked by the presence of LTRs flanking the central protein coding region. These long direct terminal repeats, containing approximately equals 250 to 600 base pairs, are characteristic of integrated retroviral DNA and are critical to the life cycle of retroviruses. In addition to sharing LTRs with retroviruses, LTR retrotransposons encode all the proteins of the most common type of retroviruses, except for the envelope proteins. Lacking these envelope proteins, LTR retrotransposons cannot bud from their host cell and infect other cells. However, they can transpose to new sites in the DNA of their host cell. A key step in the retroviral life cycle is formation of retroviral genomic RNA from integrated retroviral DNA. This process serves as a model for generation of the RNA intermediate during transposition of LTR retrotransposons. As depicted in Figure 5, the leftward retroviral LTR functions as a promoter that directs host cell RNA polymerase II to initiate transcription at the 5' nucleotide of the R sequence. After the entire downstream retroviral DNA has been transcribed, the RNA sequence corresponding to the rightward LTR directs host cell RNA processing enzymes to cleave the primary transcriptin adepoly A tail at the 3' end of the R sequence. The resulting retroviral RNA genome, which lacks a complete LTR, is packaged into a virion that buds from the host cell. After a retrovirus infects a cell, reverse transcription of its RNA genome by the retrovirus-encoded reverse transcriptase yields a double-stranded DNA containing complete LTRs, Figure 6. Integrase, another enzyme encoded by retroviruses that is closely related to the transposase of some DNA transposons, uses a similar mechanism to insert the double-stranded retroviral DNA into the host cell genome. In this process, short direct repeats of the target site sequence are generated at either end of the inserted viral DNA sequence. As noted above, LTR retrotransposons encode reverse transcriptase and integrase. By analogy with retroviruses, these mobile elements are thought to move by a copy and paste mechanism whereby reverse transcriptase converts an RNA copy of a donor site element into DNA, which is inserted into a target site by integrase. The experiments depicted in Figure 6 provided strong evidence for the role of an RNA intermediate in transposition of Ti elements. Sequencing of the human genome has revealed that the most common LTR retrotransposon-related sequences in humans are derived from endogenous retroviruses, ERVs. Most of the 443,000 ERV-related DNA sequences in the human genome consist only of isolated LTRs. These are derived from full-length pruviral DNA by homologous recombination between the two LTRs, resulting in deletion of the internal retroviral sequences. Retrotransposons that lack LTRs move by a distinct mechanism. The most abundant mobile elements in mammals are retrotransposons that lack LTRs, sometimes called non-viral retrotransposons. These moderately repeated DNA sequences form 2 classes in mammalian genomes: long interspersed elements, lines, and short interspersed elements, signs. In humans, full-length lines are approximately equals 6 kilobits long, and signs are approximately equals 300 BP long. Repeated sequences with the characteristics of lines have been observed in protozoans, insects, and plants, but for unknown reasons they are particularly abundant in the genomes of mammals. Signs also are found primarily in mammalian DNA. Large numbers of lines and signs in higher eukaryotes have accumulated over evolutionary time by repeated copying of sequences at a few positions in the genome and insertion of the copies into new positions. Although these mobile elements do not contain LTRs, the available evidence indicates that they transpose through an RNA intermediate. Lines. Human DNA contains 3 major families of line sequences that are similar in their mechanism of transposition, but differ in their sequences, L1, L2, and L3. Only members of the L1 family transpose in the contemporary human genome. Line sequences are present at approximately equals 900,000 sites in the human genome, accounting for a staggering 21% of total human DNA. The general structure of a complete line is diagrammed in Figure 8. Lines usually are flanked by short direct repeats, the hallmark of mobile elements, and contain 2 long open reading frames, ORFs. ORF1, approximately equals 1 kilobit long, encodes an RNA binding protein. ORF2, approximately equals 4 kilobits long, encodes a protein that has a long region of homology with the reverse transcriptases of retroviruses and viral retrotransposons, but also exhibits DNA endonuclease activity. Evidence for the mobility of L1 elements first came from analysis of DNA cloned from humans with certain genetic diseases. DNA from these patients was found to carry mutations resulting from insertion of an L1 element into a gene, whereas no such element occurred within this gene in either parent. About 1 in 600 mutations that cause significant disease in humans are due to L1 transpositions or sign transpositions that are catalyzed by L1 encoded proteins. Later experiments similar to those just described with yeast tie elements, see Figure 6, confirmed that L1 elements transpose through an RNA intermediate. In these experiments, an intron was introduced into a clone mouse L1 element, and the recombinant L1 element was stably transformed into cultured hamster cells. After several cell doublings, a PCR-amplified fragment corresponding to the L1 element but lacking the inserted intron was detected in the cells. This finding strongly suggests that over time the recombinant L1 element containing the inserted intron had transposed to new sites in the hamster genome through an RNA intermediate that underwent RNA splicing to remove the intron. Since lines do not contain LTRs, their mechanism of transposition through an RNA intermediate differs from that of LTR retrotransposons. ORF1 and ORF2 proteins are translated from a line RNA. In vitro studies indicate that transcription by RNA polymerase II is directed by promoter sequences at the left end of integrated line DNA. Line RNA is polyadenylated by the same post-transcriptional mechanism that polyadenylates other mRNAs. The line RNA then is transported into the cytoplasm, where it is translated into ORF1 and ORF2 proteins. Multiple copies of ORF1 protein then bind to the line RNA, and ORF2 protein binds to the poly A tail. The line RNA is then transported back into the nucleus as a complex with ORF1 and ORF2. ORF2 then makes staggered nicks in chromosomal DNA on either side of any A-TRH sequence in the genome, Figure 9, Step 1. Reverse transcription of line RNA by ORF2 is primed by the single-stranded T-rich sequence generated by the nick in the bottom strand, which hybridizes to the line poly A tail, Step 2. ORF2 then reverse transcribes the line RNA, Step 3, and then continues this new DNA strand, switching to the single-stranded region of the upper chromosomal strand as a template, Steps 4 and 5. Cellular enzymes then hydrolyze the RNA and extend the 3' end of the chromosomal DNA top strand, replacing the line RNA strand with DNA, Step 6. Finally, 5' and 3' ends of DNA strands are ligated, completing the insertion, Step 7. These last steps, 6 and 7, probably are catalyzed by the same cellular enzymes that remove RNA primers and ligate Okazaki fragments during DNA replication. The complete process results in insertion of a copy of the original line retrotransposon into a new site in chromosomal DNA. A short direct repeat is generated at the insertion site because of the initial staggered cleavage of the two chromosomal DNA strands, Step 1. The vast majority of lines in the human genome were truncated at their 5' end, suggesting that reverse transcription terminated before completion and the resulting fragments extending variable distances from the poly A tail were inserted. Because of this shortening, the average size of line elements is only about 900 base pairs, whereas the full-length sequence is approximately equals 6 kilobits long. In addition, nearly all the full-length elements contain stop codons and frameshift mutations in ORF1 and ORF2. These mutations probably have accumulated in most line sequences over evolutionary time. As a result of truncation and mutation, only approximately equals 0.01% of the line sequences in the human genome are full-length with intact open reading frames for ORF1 and ORF2, approximately equals 60 to 100 in total. Signs, the second most abundant class of mobile elements in the human genome, signs constitute approximately equals 13% of total human DNA. Varying in length from about 100 to 400 base pairs, these retrotransposons do not encode protein, but most contain a 3' A T-rich sequence similar to that in lines. Signs are transcribed by RNA polymerase III, the same nuclear RNA polymerase that transcribes genes encoding tRNAs, 5' srRNAs, and other small stable RNAs. Most likely, the ORF1 and ORF2 proteins expressed from full-length lines mediate transposition of signs by the retrotransposition mechanism depicted in Figure 9. Signs occur at about 1.6 million sites in the human genome. Of these, approximately equals 1.1 million are ALU elements, so named because most of them contain a single recognition site for the restriction enzyme ALU-I. ALU elements exhibit considerable sequence homology with and may have evolved from 7s lRNA, a component of the signal recognition particle. This abundant cytosolic ribonucleoprotein particle aids in targeting certain polypeptides, as they are being synthesized, to the membranes of the endoplasmic reticulum. ALU elements are scattered throughout the human genome at sites where their insertion has not disrupted gene expression, between genes, within introns, and in the 3' untranslated regions of some mRNAs. For instance, 9 ALU elements are located within the human beta-globin gene cluster. The overall frequency of L1 and sign retrotranspositions in humans is estimated to be about 1 new retrotransposition in variate individuals, with approximately equals 40% being L1 and 60% signs, of which approximately equals 90% are ALU elements. Similar to other mobile elements, most signs have accumulated mutations from the time of their insertion in the germline of an ancient ancestor of modern humans. Like lines, many signs also are truncated at their 5' end. Table summarizes the major types of interspersed repeats derived from mobile elements in the human genome. In addition to the mobile elements listed in table, DNA copies of a wide variety of mRNAs appear to have integrated into chromosomal DNA. Since these sequences lack introns and do not have flanking sequences similar to those of the functional gene copies, they clearly are not simply duplicated genes that have drifted into non-functionality and become pseudogenes, as discussed earlier. Instead, these DNA segments appear to be retrotransposed copies of spliced and polyadenylated processed mRNA. Compared with normal genes encoding mRNAs, these inserted segments generally contain multiple mutations, which are thought to have accumulated since their mRNAs were first reverse transcribed and randomly integrated into the genome of a germ cell in an ancient ancestor. These non-functional genomic copies of mRNAs are referred to as processed pseudogenes. Most processed pseudogenes are flanked by short direct repeats, supporting the hypothesis that they were generated by rare retrotransposition events involving cellular mRNAs. Other moderately repetitive sequences representing partial or mutant copies of genes encoding small nuclear RNAs, snRNAs, and tRNAs are found in mammalian genomes. Like processed pseudogenes derived from mRNAs, these non-functional copies of small RNA genes are flanked by short direct repeats and most likely result from rare retrotransposition events that have accumulated through the course of evolution. Enzymes expressed from a line are thought to have carried out all these retrotransposition events involving mRNAs, snRNAs, and tRNAs. Mobile DNA elements probably had a significant influence on evolution. Although mobile DNA elements appear to have no direct function other than to maintain their own existence, their presence probably had a profound impact on the evolution of modern-day organisms. As mentioned earlier, about half the spontaneous mutations in Drosophila result from insertion of a mobile DNA element into or near a transcription unit. In mammals, however, mobile elements cause a much smaller proportion of spontaneous mutations, approximately equals 10% in mice and only 0.1–0.2% in humans. Still, mobile elements have been found in mutant alleles associated with several human genetic diseases. In lineages leading to higher eukaryotes, homologous recombination between mobile DNA elements dispersed throughout ancestral genomes may have generated gene duplications and other DNA rearrangements during evolution. For instance, cloning and sequencing of the beta-globin gene cluster from various primate species has provided strong evidence that the human G-gamma and A-gamma genes arose from an unequal homologous crossover between two L1 sequences flanking an ancestral globin gene. Subsequent divergence of such duplicated genes could lead to acquisition of distinct, beneficial functions associated with each member of a gene family. Unequal crossing over between mobile elements located within introns of a particular gene could lead to the duplication of exons within that gene. This process most likely influenced the evolution of genes that contain multiple copies of similar exons encoding similar protein domains, such as the fibronectin gene. Some evidence suggests that during the evolution of higher eukaryotes, recombination between interspersed repeats and introns of two separate genes also occurred, generating new genes made from novel combinations of pre-existing exons, figure 10. This evolutionary process, termed exon shuffling, may have occurred during evolution of the genes encoding tissue plasminogen activator, the NOI receptor, and epidermal growth factor, which all contain an EGF domain. In this case, exon shuffling presumably resulted in insertion of an EGF domain encoding exon into an intron of the ancestral form of each of these genes. Both DNA transposons and line retrotransposons have been shown to occasionally carry unrelated flanking sequences when they insert into new sites by the mechanisms diagrammed in Figure 11. These mechanisms likely also contributed to exon shuffling during the evolution of contemporary genes. In addition to causing changes in coding sequences in the genome, recombination between mobile elements and transposition of DNA adjacent to DNA transposons and retrotransposons likely played a significant role in the evolution of regulatory sequences that control gene expression. As noted earlier, eukaryotic genes have transcription control regions, called enhancers, that can operate over distances of tens of thousands of base pairs. Transcription of many genes is controlled through the combined effects of several enhancer elements. Insertion of mobile elements near such transcription control regions probably contributed to the evolution of new combinations of enhancer sequences. These in turn control which specific genes are expressed in particular cell types and the amount of the encoded protein produced in modern organisms. These considerations suggest that the early view of mobile DNA elements as completely selfish molecular parasites misses the mark. Rather, they have likely contributed profoundly to the evolution of higher organisms by promoting 1. The generation of gene families via gene duplication, 2. The creation of new genes via shuffling of pre-existing exons, and 3. Formation of more complex regulatory regions that provide multifaceted control of gene expression. D. Cell division and cell cycle. I. Systems of cell division. 1. 4 events occur before and during cell division. A signal to reproduce must be received. Replication of DNA and vital cell components must occur. DNA must distribute to the new cells. The cell membrane and cell wall in some organisms must separate the two new cells. 2. Prokaryotes divide by fission. 1. Prokaryotic cells grow in size, replicate DNA, and divide into two new cells. This process is called fission. Escherichia coli, a bacterium, simply divides as quickly as resources permit. At 37 OC, this is about once every 40 minutes. When resources are abundant, E. coli can divide every 20 minutes. Prokaryotes generally have just one circular chromosome. The E. coli chromosome is 1.6 mm in diameter, making the unfolded circle 100 times greater than the size of the cell. The molecule is packaged by folding in on itself with the A to basic proteins that associate with the acidic DNA. Circular chromosomes appear to be characteristic of all prokaryotes. 3. The prokaryotes have a site called ORI, where DNA replication begins, and a site TER, where it ends. ORI is short for origin of replication. TER is short for terminus of replication. 4. As DNA replicates, each of the two resulting DNA molecules attaches to the plasma membrane. As the bacterium grows, new plasma membrane is added between the attachment points, and the DNA molecules are moved apart. 5. Cytokinesis, which is cell partitioning, begins around 20 minutes after chromosome duplication is completed. A pinching of the plasma membrane to form a constricting ring separates the one cell into two, each with a complete chromosome. A tubulin-like fiber is involved in the persistring constriction. 3. Eukaryotic cells divide by mitosis or meiosis. 1. All reproduction involves reproduction signals, DNA replication, segregation, and cytokinesis. 2. Unlike prokaryotes, eukaryotic cells do not constantly divide whenever environmental conditions are adequate, although unicellular eukaryotes do so more often than the cells of multicellular organisms. Some differentiated cells of multicellular organisms rarely or never divide. Signals to divide are related to the needs of the entire organism, not simply the opportunity created by resources. 3. Eukaryotes usually have many chromosomes. Eukaryotes have a nucleus, which must replicate and, with few exceptions, divide during cell division. Mitosis generates two cells with the same genetic information as the original cell. 4. Meiosis is a specialized cell division used for sexual reproduction. The genetic information of the chromosomes is shuffled, and the cells, called gametes, typically get one half of the original DNA complement. D1. Mitosis. Mitosis is the process by which a eukaryotic cell separates the chromosomes in its cell nucleus into two identical sets in two daughter nuclei. It is generally followed immediately by cytokinesis, which divides the nuclei, cytoplasm, organelles and cell membrane into two daughter cells containing roughly equal shares of these cellular components. Mitosis and cytokinesis together define the meiotic phase of the cell cycle, the division of the mother cell into two daughter cells, genetically identical to each other and to their parent cell.